Thanks everybody for joining. Looks like the attendee increase has slowed down a little bit, maybe a few trickle in, but we'll get started with an introduction. So today we have, this is the second installment of the BioNano webinar series, and we have a great speaker for us today, Dr. Jaroslav Dolezel. He's at the Institute of Experimental Botany in the Czech Republic. Jaroslav has a really great project to share with us today, where he's used our technology downstream of flow sorting wheat chromosomes. So really putting together his expertise along with the extremely long molecule, single molecule imaging of the Irys system to get a very unique perspective on the wheat genome. Before we get into Jaroslav's presentation, however, I'd like to give a brief introduction to the technology and product so everybody's on the same page and understands how Jaroslav put it to good use in the wheat project. So I'll introduce myself first. My name is Harper Van Steenhouse. I'm the market manager here at BioNano Genomics, and I'll just share with you a few slides describing the Irys platform and the technology that underlies it. So here at BioNano Genomics, we are all about giving you a different kind of view on the genome. Here's a movie showing you this kind of view that we're talking about. This is a single molecule of DNA, 400 kilobases long, being separated and loaded into nanochannels to do single molecule imaging. So this is a movie, just a brief introduction of what the technology has in store, but I'll share with you much more depth as we go through. So with this single molecule, extremely long DNA view of the genome, it enables a variety of solutions. We're able to generate higher quality de novo assembled genomes much faster and less expensive than using old techniques such as back cloning and such. We're able to generate much more meaningful biological information because we're doing single molecule imaging at a very high resolution. In addition, structural variation is captured directly. Oh, sorry. Just got a question in. Kim, did you see? Okay. Sorry, a little technical difficulty here. Okay, we're back. Center? We got a couple of notices. Some people are having a hard time hearing us. If somebody could enter into the chat box whether they can hear me now, that'd be great. Okay, some people can hear us. If you're having a hard time hearing us, perhaps, okay, yeah, they can't hear us if they won't hear those instructions. Okay, so it looks like everybody's hearing us now. Sorry for that technological glitch here. So, as I was saying, really the underlying benefit here is the ability to measure on a different scale than sequencing technologies do. So rather than looking at individual base pairs, we're looking at extremely long molecules on the resolution of kilobases to megabases. And to get this better view, we retain long-range contiguity so you can see repeats and structural variation directly by virtue of not fragmenting the DNA, stretching it out and linearizing it in a uniform fashion to get very high precision and reproducible measurements, and using this long molecule view of the genome so that we can use this higher sequence uniqueness to assemble complex genomes at a very high throughput fashion. So when you have extremely long molecules, you end up with extremely long assemblies. This is just an example of a collaboration where we took 12 megabase genome and we're able to assemble it in a single contig into a physical map of its predicted 12 megabase size. And this is something you can't do with short read technologies, even when you include long reads or long paired information with it. So by virtue of having this information, you're able to get a view of structural variation, again, on the order of kilobases and megabases, which are the size, the structural variants that you're looking for. In addition, a lot of what Jaroslav is going to be talking about today is getting into scaffolding and assembly validation of existing sequencing work, sizing gaps and or resizing or confirming gaps, and also by virtue of the single molecule technology, pulling apart haplotypes and seeing differences between the two haplotypes of genomes. So we've used this technology, and our customers have used this technology in a variety of different organisms. Certainly human is one area of focus, but things like crops, fungi, pests, bacteria, and other model organisms have all been tackled with this technology to great success. So I've given you a little bit of information about what you can do with the technology. Now I want to just dive in a little bit more of how it works and what this product really is. So Irys as a platform includes both the instrument that automates the processing and imaging of the Irys chip. The Irys chip is where you load your sample that has been prepared using the Irys Prep reagent kit, and then results are captured and analyzed in the Irys View software suite. The key to getting these very long molecules, which you saw a movie of already, is confining single molecules in as native state as possible in very small channels. What this does is allows you, the view, to capture them and hold them still and detect the labels that you put in them with very high precision. It's not easy to get these DNA molecules into nanochannels, however, and that's the function of the Irys chip, to separate DNA while it's stuck together in free solution, get it linearized and in single molecule fashion into its own nanochannel for imaging. The Irys chip version 2 is the high throughput version of our chip. It allows you to do it allows you to do a human genome to 30x depth de novo using 3 chips. There are 2 flow cells on each Irys chip, which enable you to split up smaller genomes across chips and gives you the flexibility of how to run it. And each of those Irys chips take 1 day to run. On each flow cell of the Irys chip, there's 13,000 nanochannels, and this is where you can see the bottom right, you can see the SEM showing you these 45 nanometer nanochannels where the DNA are captured and imaged. I'll show you a movie of how this looks across a number of channels soon. The first step, as I mentioned, is a sample preparation. This is where, using the Irys Prep reagents and protocol, you isolate extremely long DNA and label in a sequence-specific fashion. The labeling occurs as described here. The first kit that we offer is based on a nicking enzyme, which recognizes a 7-mer sequence throughout the whole genome, puts a single-strand nick at each of those, and then we use a polymerase and a fluorescently labeled nucleotide to incorporate at those sites. And your resultant sample is a very long pieces of DNA with green labels at every instance of a 7-mer. The picture on the right show you what this looks like now. 2-color image of DNA molecules in channels. These are each extremely long molecules shown separated in their own nanochannels with a series of dots, green dots on the blue molecules. These green dots represent the 7-mer sequence that the nicking enzyme recognized and establishes essentially a barcode in the DNA so that it can be uniquely assembled to in an overlap layout consensus fashion or aligned back to a genome of interest. Here's the movie I told you that would demonstrate what this looks like as molecules are loaded into nanochannels, moving through a gradient of decreasing sized micro and nanostructures to separate them out and get individual molecules in nanochannels. first you see DNA moving into pillars, and as they continue to be electrophoresed from left to right in this image, moving into microchannels, and ultimately nanochannels, where single molecules of DNA are separated and linearized for imaging. This movie will loop through several times, but each instance of this represents one cycle of loading nanochannels in the Irys chip, followed by automated imaging of these nanochannels. And then the instrument simply pulls another cycle of DNA through and does imaging again to generate extremely high throughput from a single sample on a single chip. That's depicted here schematically, where you see images collected over and over again of new molecules from the same sample. The software will then detect where these molecules are and where the labels are on the molecules, digitize that information into individual molecules where the assembly algorithm can step in and perform the assembly de novo using the patterns to generate a very accurate consensus map, which essentially represents a physical map of your genome that you ran. Once you have this physical map, you can then compare it to other maps. Depending on your application, you may want to compare it to a reference and look for differences that would represent structural variation. You may want to compare it to another individual of the same species and look for variants. Or you may want to load in your sequencing information, contigs or scaffolds, and use that to order and orient them to the genome map that's created and do some scaffolding or super scaffolding. Structural variations look like as depicted here. It's essentially the absence or presence of extra labels in one map versus another, or labels that get closer together or further apart relative to another map. This is done without the reference. The sample is assembled completely without bias from the reference, so that you don't incorporate any reference bias as you would get it with arrays. And you're able to detect a wide range of sizes down to single-digit KB all the way up to megabases. I mentioned earlier that haplotyping is feasible by virtue of the single molecule detection. That's described here. As you'll see, the sequence assembly, as many are in green, are assembled into a single haplotype representation of a diploid genome, whereas the individual molecule reads coming off of Irys depict each haplotype separately and can be assembled into two separate maps shown in these two different blue. And you'll see the differences exist within this blue box, whereas outside the two haplotypes are identical. So with that, I'm about to hand it off to Jaroslav to demonstrate one example of how this technology can be used. But just to remind you, the Irys platform is used in a variety of cases for a number of applications where high throughput, long-range information is essential to do direct measurement using a single molecule imaging to look at genome architecture, organization, and context that really are hard or impossible to detect using existing methods. And with that, a number of groups are looking at structural variation as well as doing higher quality de novo assembly than they've ever been able to do before. We'll have time for Q&A for Jaroslav at the end of his presentation, so please enter your questions into the chat box. Also, if you have any additional questions after the webinar, please feel free to contact us at sales@bionanogenomics.com. You can check out our webpage at bionanogenomics.com, and we'd certainly love it if you followed us on Twitter. Okay, with that, I'll hand it off to Jaroslav.
And welcome to everybody. Good afternoon if you are in Europe. Thanks for introduction. My name is Jaroslav Dolezel. I work at the Institute of Experimental Botany in Olomouc, Czech Republic. As you know, our project, as many of you may know, our projects are focusing on facilitating the analysis of large and complex plant genomes. So I'm very happy that I can share some of our results obtained with applying bio-nanotechnology. In the first part of my short presentation, I will. Sorry?
Hi, Jaroslav, it's Harper. We need you to share your slides.
Sorry?
Yeah, share your screen or desktop.
Yeah. I'm not sure what to do.
If you're in the WebEx Event Center,
yes.
You'll see a button down at the bottom saying Share Applications or Share Desktop.
Yeah. Is it okay now?
That.
Is it okay?
Yes. Yes. Thank you very much. Sorry to interrupt.
I'm sorry, I overlooked that. All right. So at least you could probably hear me. I was not showing anything important, so I perhaps can continue. So, well, what I like to do today is to briefly mention the problems with sequencing large and complex plant genomes. And in fact, our research activities are focusing on facilitating the analysis of these genomes, and that's the reason we've been pioneering the approach we called chromosome genomics. So I will briefly introduce this concept, and later on I will describe how we can incorporate, how we can use the technology developed by bio-nano to facilitate the assembly of these complex genomes, and also I'd like to share our plans for the future regarding the application of this technology. So very briefly, we all are well aware of the rapid spread of next generation sequencing technologies, and one would expect that there will be a number of species sequenced to a high quality. That's not really true, and the general observation is that the quality of de novo genome sequences was rather decreasing, and in fact this is the consequence of routine application of next generation sequencing technologies. There were a few papers published recently, last year, this year, who point to this problem, and this is nicely shown in this small cartoon, which compares the small part of the rice genome with the sequence obtained using the old style back-by-back sequencing and using the shotgun next generation sequencing, where you see many problems with the assembly. So the bottleneck is definitely not the sequencing technologies, just the opposite. The capacity is increasing. The problem is how to assemble short sequence reads in genomes where the repeated DNA is prevalent, and in genomes which are polyploid. There are estimates that read lengths exceeding 200 kb may be needed in some very complex genome in order to be able to unpick repeats and other complications. Current sequencing technologies fall short of this, obviously. So there have been many strategies which were suggested and employed to improve genome assemblies. One of them is ultra high depth sequencing, phosphate pooling, where you sequence smaller fractions of the genome randomly selected, long fragment read technology, which simply aim to achieve that goal of having long reads and facilitating the assembly. This obviously includes much expected Oxford nanopore technology, but there comes optical mapping or mapping on nanochannel arrays, as presented by Harper and developed by bio-nano, which really has a chance to reach long regions of genome which may be complex structure. And last but not least, the idea, the approach may be to reduce the sample complexity by for example, sequencing only single chromosomes. So in this case, the genome complexity is reduced to a small part of the genome, and the assembly is facilitated, and obviously also the time needed for that is shorter. So this presentation covers these two areas: the optical mapping, mapping on NanoChannel arrays, and reduction of sample complexity. Now, the reduction of sample complexity by sequencing single chromosomes is the focus of research in our laboratory, and the application of the sequencing isolated chromosomes is called chromosome genomics. So the chromosome genomics uses all tools of genomics, but applies them to single chromosomes or even the small parts, which may be, for example, the arms. The application of this technology is nicely demonstrated in wheat. The wheat genome, the bread wheat genome, is huge as compared, for example, to rice, with almost 17 giga. The problem with working with wheat is not only the size of the genome, but also the fact that it's a hexaploid species which consists, in fact, from 3 subgenomes, each of them comprising 7 chromosomes. So we deal with 3 different genomes which are similar. So the sequences coming from chromosome 1A, 1B, and 1D are homologous. They are similar and may be difficult to discriminate. Now, by isolating a single chromosome, not only I reduce the complexity of the sample to smaller part, but I'm also avoiding the problem due to polyploidy. So by working with single chromosome, we reduce the complexity to 3 to 6% of the whole genome. By working with single chromosome arms, as these can be sorted from some lines in wheat, we go down to only 1 or 3% of the genome, and working with only 700 megabase pairs. So the reduction of sample complexity is the big advantage, and avoidance of polyploidy, the problems due to sequence redundancy and polyploidy, has really big advantage. Now, this approach, the chromosome genomic, has been applied or adopted by the International Wheat Genome Sequencing Consortium, who decided to coordinate global work on development or establishment of physical map of the whole wheat genome, chromosome by chromosome or chromosome arm by chromosome arm. Now, this cartoon shows most of the chromosomes in green color, meaning that the physical maps are completed or close to completion. Individual flags indicate different countries and laboratories which are responsible for constructing physical maps of all of these individual chromosomes. Now, the consortium also initiated a project on shotgun sequencing in flow-sorted isolated chromosomes and chromosome arms. This is the second line of this cartoon, and as you see, all chromosomes were already sequenced. Most of them sequenced by the consortium, some of them sequenced by individual laboratories with own funding. Obviously, the final goal is to develop pseudomolecules, so develop a reference sequence of the wheat genome, chromosome by chromosome. And once again, you see the flags indicating the laboratories and countries responsible for that. You may notice that so far, 1 chromosome has been completely sequenced. This is chromosome 3B, and this work was done in France, and coordinated by Catherine Foyer and led by Frédéric Choulet from INRA in Clermont-Ferrand. This is a beautiful piece of work showing that even such a complex genome can be sequenced using the assembly of current technologies and applying the chromosome genomics approach. Imagine or remember that the chromosome 3B is almost 1 gigabase pair long, so it's 1/3 of the human genome. This chromosome was chosen as for a pilot study. As I mentioned, the physical map was developed, and the hybrid approach to sequence the chromosome was applied. So the next generation technology was applied, but was applied in combination with the physical map and the back-contig physical map. In this case, the backend sequences were obtained, minimum tiling path back clones were sequenced in pools by 454, but on the top of it, the chromosomal DNA was shotgun sequenced by Illumina. So it's a combination of different approaches. which resulted in an excellent assembly, high-quality assembly consisting of about 3,000 scaffolds, with representing most of the chromosome 3b, which over 800 mega, and N50 of close to 900 kb. This paper should be soon published by Fred Schulet and colleagues. Given that, we see that there's a chance to develop a high-quality de novo reference sequence of a genome, but yet still there are gaps in the assembly, and we still may need some other technologies to complement the existing technologies. And I'm convinced, and many other colleagues as well, that the physical mapping using the NanoChannel technology of BioNano can do the job. That was the reason we decided to test this approach in collaboration with colleagues in BioNano. And we choose a chromosome arm, short arm of chromosome 7D of wheat. This arm is about 400 mega long and represents 2% of the genome. So it's a very small part of the wheat genome, and we chose it because my laboratory is responsible for sequencing this arm. This arm was purified by flow sorting. This is a flow karyotype of a double D-telosomic line of wheat showing the peak of chromosome arm 7DS, which was flow sorted, purified, 7DS, 7DL. And the reason we choose this arm is that we had a number of resources available, molecular resources available for this chromosome arm. First, the chromosome was shotgun sequenced by Illumina, and we identify about 1,700 anchored gene loci. We have a physical map ready, which consists of 900 contigs with N50 520. The map was done using both FPC and LTC software. And the important point for the decision was that the Illumina sequencing of MTP back clones 4600 is in progress. This is the collaboration with Dave Edwards and Jackie Butley in the University of Brisbane. The availability of the shotgun sequences allowed us to check, to verify the nicking enzymes that would be suitable for this work, developing of the nano map. And this analysis clearly showed that we may expect about 13 sites per 100 kb using BSPQ1, and this was chosen for the following experiment. As chromosome sorting or sorting chromosome arm in large numbers may be laborious, we wanted to test the suitability of chromosomal DNA using a simple approach. So we flow sorted all wheat chromosomes just to have chromosomal DNA available, and we wanted to check whether the chromosomal DNA is suitable for this type of application. Remember, the critical is the quality of DNA, the length of DNA molecules. So we flow sorted all wheat chromosomes, 300 chromosomes per miniplug. The miniplug's about 20 microliters, and we use pulse field gel electrophoresis to check the quality of the DNA. So the first point was to see if the DNA is intact. And in fact, what we observed was that the majority of DNA remains in sample wells, even after treatment with proteinase at a high concentration and long treatments. At the same time, it's important that this DNA is accessible. So we wanted to see how the DNA is digested using a restriction enzyme. And once again, the results were very positive. We observed that even with low concentration of restriction enzyme and a short time of digestion, the DNA was practically all digested. There was nothing left in the sample well. So with this, we were quite convinced that the flow sorted chromosomes are indeed suitable for mapping by Irys, BioNano. The quality of DNA was high. Based on the analysis by BioNano, the N50 was about 100 kb, and once again, the expected label density with BSPQ1 was really reaching the theoretical 13.100 kb as indicated in this cartoon. So at this point, we were ready to work with the 7DS arm. We flow sorted the 7DS arm and embedded this chromosome arm in 3 mini plugs, so in total we sorted about 1 microgram of DNA. These plugs were used to recover the DNA. The efficiency was rather high. We recovered about a half microgram DNA. The DNA was labeled and analyzed by BioNano using the version 2 chip. The results were very nice. This is the cartoon showing the distribution of the length of DNA molecules recovered from flow-sorted chromosome arm 7DS. And as you see, there were molecules exceeding 2 million base pairs. So that was quite nice. And this small table shows you the statistics. There was a threshold set for 150 kb that was used then to assemble the optical map. With this threshold, we had about 19K molecules with total lengths of 60 giga, 200x coverage of the arm, and 5344 kb. Now these molecules were used to assemble the optical map. So they're collected at 200x read depth. In total, 371 genome maps were de novo assembled. The total length of assembly is 350 mega. Remember, the length of 7DS is 380. So we obtained the map of 7DS, which covers 92% of the estimated size. The average map size is 0.9 mega, and N50 is 1.3 mega. And this cartoon shows you one of the genome maps. So that's one of the fragments of the genome which were assembled using the NanoChannel array. And you see in this case, the map exceeds 2.8 mega bases, which is really extremely nice, extremely long. And well, then you see individual molecules, single molecule maps, and that. Well, it's maybe interesting to see that there are long gaps where the restriction fragments are missing, and yet they can be breached. They can be breached. They can exceed 100 kb. They can be breached simply because you work with long, extremely long molecules of DNA. Now, everybody working with plant genome knows the problem with genome assembly, sequencing and assembling, analyzing repetitive DNA. And tandem repeats, long tandem arrays are really a nightmare to assemble. And so it's interesting to see how this approach can deal with them. This cartoon shows you the frequency of different types of tandem repeat units, if you wish, with differing in different length of the unit and indicating the frequency of this unit. And there's a peak showing presence of a tandem repeat with about 10 kb unit. There should be about over 80 copies or 80 copies of the array. Now, this genome map 350 clearly shows the presence of such an array. The array is long as long as 850 kb, so there will be about over 80 units of these tandem repeats. This is a beautiful result and showing the ability of this approach to cover, to pick up, to characterize such repeat, which can't be done using any other technique. Now, the obvious question is how you can use the optical map to assemble DNA sequences. We are still starting, and we work intensively with Dave Edwards on assembling the sequences obtained of sequences from back clones from 7DS. So I will show you only the first results, where we in a pilot experiment we just wanted to see how we can anchor 5 overlapping back clones to the genome map. These are the back clones which are overlapping based on the physical map, and this cartoon shows you the integration of the nano array map, the BioNano Irys map, and contigs obtained after sequencing those 5 bug clones. Well, you see that they can be nicely, these are the bug clones, the contigs obtained from bug clones, the bug clones addresses. Now you see that the bug clones can be nicely anchored to the map, and there are some interesting observations. So I like to point only one or two to one of them. First of all, you may notice that the contig 5, which correspond to this bug clone, links in fact to nano maps. So we can even make the map longer. There's a part of the nano map which is not doesn't seem to be included in the assembly of the bug clone, which may indicate some problem with the assembly of this clone. So this may be one or two issues or interesting observations. We still work on improving the assembly, and my opinion is that the interaction between the nano array map and the sequences from bug clones will lead to high quality genome assemblies. Well, this arrow points to a small region where we don't have any restriction point, so we are not quite sure whether this part of the assembly is correctly done. This was probably a simple presentation showing the very first results we have obtained, but nevertheless, the conclusions are, I believe, important. First of all, flow sorted chromosomes can be used for genome mapping on nanochannel arrays. The relatively small number of chromosomes that is needed is sufficient, which is important. 1 million chromosomes can be sorted in few days. It's important that this technology seems to fulfill the expectations, as the map we have developed for 7DS ARM covers almost all entire predicted length. Finally, the shotgun sequences from bug clones can be anchored on the chromosome map. Back to timing, this is the indication, the time frame of this project, which was done, I think, in a very short time, and yet we were not really in a time pressure. So after some quality checks of all chromosomal DNA, we were flow sorting the 7DS, but as I mentioned, can be done in a much shorter time. We were simply not in time pressure. But the beauty is that, I mean, the interesting point is that in a very few days, the nano array map was developed and assembled. So in 1 week after delivering the DNA or supplying the DNA, we had the map ready. Well, what's the outlook? We are convinced that chromosome map will be useful for genome sequence validation, anchoring, and scaffolding in plants, and definitely in species which have complex genomes and polyploid genomes, and obviously bread wheat is just the prime example. The maps, we cannot wait to see the use of this technology to facilitate gap closure, validation of repeat-rich genome regions, and the analysis of copy number and structure variation at the chromosomal level, as indicated by Harper and his introductory talk. And also, I was talking about chromosomes. I like to point to the fact that as we sorted mitotic chromosomes, obviously we can do the same with interface nuclei. So this approach I have presented is not limited to chromosomes and can be used in other species where either we can't sort chromosomes or we are not interested to work on a chromosome basis, and we can simply purify interface nuclei and construct the maps for the whole genome. The advantage of this approach would be that we obtain highly pure fractions of nuclear DNA, and the DNA will be highly intact because we sort intact nuclei with high molecular weight DNA. At this point, I'd like to stop, and I'd like to acknowledge the people who contributed to this success. First of all, my group in Institute of Experimental Botany in Olomouc, Hanna Šimková, who was really very helpful and did most part of the work in preparing the high molecular weight DNA, Jan Vrana and Maria Kubaláková, who did all the work with chromosome sorting, Helena Staňková, who was helping a lot in assembling the back sequences to the nanoarray map, and Romana Špekova for technical support. This work would not be possible, and I'm so grateful to people in BioNano, Alex Hesty and Harper Van Steenhuis especially, who were so communicative, who understood the differences between classical DNA samples and flow-sorted chromosomes, and also Jackie Chen and Han Kao. This work on 7DS in particular relies a lot on collaboration with Dave Edwards and Jackie Betley at the University of Queensland in Brisbane. Dave and Jackie are so kind, they agreed to collaborate with us on sequencing the NTP, so they've been sequencing by Illumina back pools from these chromosomes. This work was done as within the framework of the International Wheat Genome Sequencing Consortium led by Kelly Eversole, and the consortium is very much interested to apply this approach to all chromosomes of wheat to facilitate genome assembly and improve it. The older work on chromosomes on wheat was as always supported by Catherine Foy, now with Bayer, but before in INRA. And the work on 3B in particular was done by Etienne Poe, who was responsible for developing physical map, and Fred Shellet, who is responsible for sequencing, and I showed parts of the results. Because we work on 7DS, it's the D genome of wheat. We collaborate a lot with Jan Dvořák and Ming-Cheng Luo in University of California, Davis. And the collaboration is very fruitful as we can exchange the information about the wheat D genome sequence and the sequence of wheat D genome progenitor Agilopstauchii. So this work was always important. Last but not least, the sorting of 7DS was possible because we obtained, or we received seeds of double heterosomic line of Chinese spring. It's a cultivar of wheat which carries the chromosome 7DS, 7D in a form of two telocentric chromosomes representing the both arms, the 7DS and 7DL. So this is greatly appreciated. Now, obviously, the support I received and been receiving from all funding agencies. And I thank you for your attention. I'm happy to discuss these results. Thank you.
Great. Thanks, Jaroslav. This is Harper again. We have one question that came in regarding technology, but I'll remind everybody, go ahead and type your questions into the chat box for Jaroslav, and I'll read them off to Jaroslav. The first one I think is a question for BioNano, so I'll address it first. The question is regarding whether we sequence directly these large DNA fragments that are separated using BioNano technology. And the answer is currently no. The DNA flows out the other end of the nanochannels and are not individually captured for sequencing. But it's a very common question and a very exciting suggestion for sure. We agree. So while maybe people are typing in questions, I have a question for you, Jaroslav. You mentioned the option both to look at flow-sorted chromosomes and whole nuclei, so whole genomes. I wonder if you could share with us your thinking about when you would use either of these two methods. Would you use them in combination, or are specific instances where you'd pick and choose between the different approaches?
Well, there may be several points to consider. The first, the one point is to see whether you need a chromosome map at all. If you need a whole genome map, and if you are able to handle the whole genome, I mean also to process the data obtained by the Irys system, then probably you can go straight away and work with the whole genome. We still need to see how far we can go, whether it is possible to develop the nanoarray map, the physical map of the whole wheat genome, or whether it is still better to go with chromosomes. But so the answer is very simple. If you can do whole genome, why not? You can do that. The nuclei will be always the best source of DNA. And remember that we saw the nuclei from specific phase of cell cycle. So they all are the same. They are not from S phase, G2 phase, they are all from G1 phase. Second is, you may be interested to analyze specific chromosomes. This will probably not be very frequent cases, but you may be interested to analyze some restructured chromosome. You like to isolate it from all other chromosomes and see specifically the structure of that particular chromosome. In that case, you will go for chromosomes definitely.
Yeah, absolutely. Makes sense. And so, and I guess the other, from our experience doing in human up to human mammalian size, certainly it makes sense to do the whole genome all at once because you're able to assemble that. So great points. We've got another question that came in. What do you expect the level of differences of high-resolution bionano map between wheat and genome donors, i.e. goat grass, A2 sheep?
Yes. I mean, if the question is whether I expect differences, yes. And I think this is one of the approaches which we should use and we want to use. We are very much interested to study the genome structure of wild wheat relatives. And this is probably one of the most attractive approaches.
Great. Had another question about the bionano product, so I guess I'll answer that. The question is, how easy is it to integrate the sequence in bionano maps? Is there software available, or is it all ad hoc? The answer is, this is a native feature of Irys View data analysis software that comes with the system. You can simply, this is where the genome maps are assembled de novo, and you can just import your sequence files as FASTA, for instance, and do the alignments of the sequence to the map, and do that work right in there. So, and then when you're done, you can certainly export it into standard formats or use Irys View just for visualization. We had a couple questions asking about transcripts or slides. I just wanted to let everybody know we are recording this and we will host that. Give us a few days time for that to get posted, but everybody will have the ability to see this later. We got a question about the cost of the chip. The list price for the Irys chip is $900. Okay. Looks like maybe some more questions coming in. In the meantime, Jaroslav, I wonder, this was a very wheat-centric talk, which is, the work that's been done so far. I wonder if you wanted to talk about your thoughts on the potential for use in other species or other areas that you're interested in.
Oh, for sure. So, yes, it was on wheat because I think this is where, if applied, this technology will make a huge impact, given the importance of wheat and given the complexity of the wheat genome. Yes, we are planning to apply this technology to many other species. You may imagine, you may consider species where with very complicated structure like saffron. Saffron crocus is a triploid species with a huge genome. It's very difficult to handle. I'd like to apply it to, or to see the application in banana. Once again, very complicated genome, not large, but clonally propagated, difficult to develop genetic maps. So physical map can be very useful to see, to study the structure, differences between different clones, etc. Yes, there are some other species where we like to apply it. So I have no doubt that we will be very busy with using this approach. And last but not least, already mentioned, relatives of cultivated wheat, and this can be extended probably to relatives some other crops as well.
Great. Does anybody else have any questions? It looks like we've answered everything that's come in the chat so far. Maybe give people 30 seconds to type if they are. Otherwise, we'll wrap up here. Okay. So with that, thank you everybody for attending. Jaroslav, thanks again for a great talk. I really appreciate it. This is a really exciting project. I'm really glad you got the opportunity to share this with all the attendees here today. So again,
thank you very much.
Yeah, thanks for sharing with the group, especially how usable this is, even with an advanced genome and all the genetic tools that you have access to with the wheat, and are still able to make progress. It's really great to see how fast this can be done, and your thoughts on how this can be really a routine method for expanding the genomics of different organisms. So thank you, everybody. Again, if you have any questions, feel free to email BioNano. We can get those to Jaroslav if you think of something after the fact, and feel free to browse our website for more information. Thanks. Have a good day, everybody.
Thank you very much. Bye.